My name is Eunice Nduati. I'm a research scientist at the Kenya Medical Research Institute Wellcome Trust Programme. I'm based at the Kilifi campus, and I'm immunologist. So my main interest is understanding host immune responses. How do our bodies respond to the things that we see or encounter? And largely, I'm working in the field of HIV. So my work largely is to understand HIV immunology and how that informs vaccine design. My main area or my research focus, or the work that my group focuses on, is really understanding immune responses, and more specifically within our population, our African population, and what are those factors that may influence how individuals within this population may respond to a vaccine. And because our field is in HIV, how do individuals respond? Are there any factors that would influence how an individual responds to a HIV vaccine within our setting? First, the key questions that we want to answer are, one, what's the composition of our immune system in terms of what's the composition of our immune repertoire? And so the sort of things we do is look at what immunoglobulin genes is an individual using, what are the populations or the frequencies of cells that we feel are important starting cells that are going to create the immune responses that we feel are relevant for protection. The other question we want to answer is, what are exposures that we see in this population that may influence the responses that an individual makes? For example, does exposure to other infections then influence how you respond to a vaccine? We know that responses in the South or in low and middle income countries have been shown to be much lower than the responses that are seen in the high income countries. So we are trying to tease out what are the mechanisms, and does exposure to some pathogens influence how you respond to this thing? The other critical question we want to understand is understand actual immune responses within clinical trials. So we have been supporting immunogenicity endpoint analyses for some clinical trials. And are there any differences how the magnitude of responses we see in our populations compared to those in Northern partner countries? And this is important because that starts telling us, can we make a global vaccine, or are there things that we need to put into consideration? So one of the research projects that we've been doing recently is that we were involved in doing the immunogenicity endpoint analysis for the first in-human vaccine trial that used what we call a germline targeting approach. And what this approach does is that it wants to prime or trigger a very specific type of cells and then teach them or shepherd them to become the sort of response that we want to make. So for this vaccine trial, what we wanted to do is to understand in our population, are people really triggering that response? The aim was not to make the functional response at the end of the whole path, but to see, can we, in the first instance, prime the correct response? And do those responses have the characteristics that we know will be able to go down the path to become the functional responses that we want? And I'm particularly really pleased with that piece of work that we did, because initially there was a lot of skepticism whether the extensive analysis of single cell analysis could be done within the continent. And I'm quite proud of the team that they were able to pull this through. And this has sort of created a lot of opportunities for other discovery work and also involvement in other clinical trials. Now that we still don't have a HIV vaccine, the big question is, can we train the human immune system to make the response that we want that will be broad enough to cover the different HIV virus clades? That's generally what we want to achieve, and also understand whether is the virus evolving in a way that the interventions that are being put in place will not work. I think the work that we do makes a difference in two fronts. One, because we are looking at informing vaccine development. It looks at healthy populations. We know that Africa bears the biggest burden of disease. And therefore, the work we're doing, if we're able to get an effective vaccine, will have a big impact in our context, whereby 65% of the burden is. I think the work also does have an impact on people who are already living with HIV, on the basis that immunotherapeutic interventions would be quite critical in the cure arena, whereby if you have individuals who are controlling viremia through being given antiretroviral drugs, then these immunotherapies can be an intervention that helps control the virus in the absence of ongoing treatment. The work we do is important. We know that HIV is still a big problem, and more so in this region of the world, which bears more than 65% of the burden. Recent reports show that more than 40 million people are living with HIV. In our setting in Kenya, we have more than 9,000 women each year being infected with HIV, and a large proportion of infants still being infected. The work that we are doing is largely to inform HIV vaccine design and is therefore critical in the bigger picture of getting an effective vaccine. The other thing that we are keen on doing is understanding how different populations respond to vaccines. It is one thing to have an effective immunogen, but it is another thing for that immunogen to be effective in a certain population. So understanding if there are other dynamics that need to be considered is quite critical. So this work, really funding the work that we are doing, is critical to the field.